So this is Matthew Reeves presenting the journal Airsac Trematodes Morcedium polonicum as a newly identified cause of death in the common blackbird. This article was published on August 2019 and its site score is 2.8. This article was published by the International Journal for Parasitology, Parasites and Wildlife. The authors include Gallosi, Maggi, and Rosi, who are affiliated with the University of Camerino in Italy, Peter Hannenberg, who is involved with Charles University in the Czech Republic, Setko, who is affiliated with Corneas Museum in Czech Republic, and Stefania Perucci, who is affiliated with the University of Pisa in Italy. In the author's introduction, they give background information on the common blackbird, also known as Turdus merula. These birds are located throughout Europe and Asia. Parasites are known to have negative effects on birds, and these negative effects include impaired fertility, weight loss, and mortality, especially among nestlings. Their goal was to use morphological and molecular analysis to identify trematode species that was found in the coelomic cavity of two deceased common blackbirds. And they also wanted to evaluate the associated pathological lesions. Two common blackbirds that were female were found dead in two different locations in Italy. One was found in January 2016 and the other was found in March 2017. About 50 adult parasites per bird were collected. Most of the parasites were fixed in a medium or loose for parasitological analysis, while the remaining parasites were fixed in 97% ethanol and used for molecular analysis. For the parasitological analysis, fixed trematodes were cleared in amine lactophenol, mounted and examined with a microscope, and were measured with the ocular micrometer. For histopathology, every organ from the coelomic cavity was collected and fixed in neutral buffered formalin for 24 hours. 2 micrometer paraffin sections were placed on a Superfrost Plus slides, and these slides were then dewaxed and stained and placed under a microscope to be observed. For molecular analysis, DNA was extracted and amplified, and primers were selected to target mitochondrial cytochrome C oxidase 1 and ND1 loci. And then the DNA underwent bidirectional Sanger sequencing using a particular DNA analyzer. The DNA sequences was then submitted to GenBank. At necropsy, both birds were heavily infected by trematodes. Gross lesions were characterized by severe colonization of air sacs, seroci, and lungs. And after the removal of the air sacs, the lung parenchyma was found congested, and there were some areas shown to be absent of gas. If you look at the picture, you can see the picture on the left. Let me draw it right here. You can see that there's this liquid to show the lack of gas. Histologically, air sac walls were covered with fibrinous exudate containing degenerate heterophiles, mononuclear cells, cellular debris, and interdispersed bacterial colonies of gram-positive cocci. As far as the parasitological examination goes, Morcedium polonicum was identified based on its morphology and dimensions, and they came to this conclusion by referencing other previously published data of Morcedium polonicum. And if you look right here on picture D, you can see the oral sucker of the parasite, and you can also on picture B right here, you can see a cluster of eggs laid by the parasite. Molecular analysis of mitochondrial and DNA loci confirmed the morphological diagnosis as revealed by the maximum likelihood approach. If you look over to the right, where their data is outlined in a box, red box, you can see it's the exact same as this one that was previously done before their experiment, but it differed from these two right here by a small increment. And for ND1, both of their sequences were the same from each bird, but differed a little bit from here, like 5 thousandths to 12 thousandths. In their discussion, they discussed how Jonah and Bland's trematode eggs were larger compared to Michalska's trematode's eggs. And this indicates that there are two different Morcillium species. But based on their morphology and measurements that they took, their species was the exact same as Michalska's, so they labeled it as Morcillium polonicum. And in their what they found in their research, that this is the first time that pathological lesions were reported and related to Morcillium polonicum. And this is the first time that death was caused by Morcillium polonicum in the common blackbird. I believe this research report is credible. It's supported by molecular analysis. It has been peer-reviewed. Their morphology of their trematode is consistent with other researchers' reports. But one of the things that would probably not make it credible is that this is the first report of pathological lesions and death caused by Morcillium polonicum in the common blackbird. So I don't know if this could be replicated.